In this last video on membrane potentials, I'm going to talk about propagation of the action potential. After you watch this video, you should be able to define propagation, it's also known as conduction, and explain why that's important. We'll also talk about a special kind of conduction called saltatory conduction. And then finally, I want you to be able to explain what myelin does and what happens if myelin is disrupted, as in what happens in things like MS, which is a demyelinating disorder. So why is it important? Well, we talked a little bit about this. If you encountered a noxious stimulus, something that's super hot, you won't move unless that information gets to the motor neurons that move the muscles. So we have a local event in the skin that's going to eventually end up in the muscles. The muscles need to understand and know that they should move. So if we have message here, something going on, somehow that information has to get into the spinal cord and then across down into the muscles so that the muscles can contract and keep you from damaging yourself further. So this process of sending that signal, as you know, the nerve impulse, the action potential is the signal. So we talked about generating that at the axon hillock. Well, once we do that, now we have to get that message that's been generated all the way to where it needs to go. That's going to be propagation. If we disrupt propagation, even though we may generate an action potential, the signal doesn't go anywhere and nothing happens. As I mentioned, we've got our typical neuron. We have our dendrites with connections, synapses to other neurons. And I do want you to remember this number, about 10,000 per neuron in the central nervous system. Axon hillock is also known as a trigger zone. This is where we first see voltage-gated sodium channels. This is where the action potential is generated. Once we sort of, the neuron, quote unquote, decides to generate an action potential, that's got to travel down the axon. And that information is then going to go into the terminal boutons, and that information will be transmitted to another neuron. One thing I want to mention right now, communication within a neuron is electrical. Okay, so that's where we're talking about membrane potentials. Between neurons is chemical. So we'll talk about the synapse neurotransmitters coming up. But within a cell, electrical. Between cells, it's chemical. Okay, so in the central nervous system of vertebrates like us, the synapse, the information will be carried from one cell to another by a chemical signal. Right? But within, it's going to be electrical. So if here we have our axon hillock, you know that voltage-gated sodium channels open up, sodium comes in, it depolarizes the membrane. At the peak of the action potential, we're at plus 30 millivolts. Somehow, again, we got to get this signal from here to here. When those ions leak in, they come in, they're going to leak to the side a little bit. Again, if we're adding positive charges, eventually we're going to reach threshold of the adjacent regions. Once we reach threshold, voltage-gated sodium channels open. So if you remember from this one, right, I've got lots of sodium. I've got a membrane depolarization here. That is then going to open the voltage-gated channels. When those open up, sodium's going to come in and cause a large depolarization. And that's what we see here. Now, the place where we were, remember the voltage-gated sodium channels become inactive. That's part of the refractory period. And then they reset and are closed. On this side, though, these are closed and ready to go. So when the ions and the depolarization of the membrane come in, now we're going to open the voltage-gated sodium channels, and sodium's going to come into the cell. Okay, so propagation or conduction is one way. It's one way because here toward the exon hillock, these voltage-gated sodium channels are refractory. These are not. They can respond. So although the ions leak both ways and affect the membrane potential on both sides, because these are refractory, they don't open up. These, because they're not refractory yet, they do open and sodium comes in. Okay. Another way of looking at it, I'm going to show you several different diagrams. One will probably click more than the others, and that's the one you should focus on. So we have our stimulus. As you know, sodium is going to come into the cell, depolarize the membrane. When that happens, we're going to then eventually trigger the voltage-gated sodium channels next to it because we're above threshold, and so on, all the way down the axon. Okay, so in this case where we're looking, we basically have voltage-gated sodium channels all along the axon. This is what happens in invertebrate axons. Invertebrates don't have myelin, okay, so they're going to have channels all the way down. In humans, most of our axons are myelinated. That has important implications that we'll get to in a second. Here's another diagram sort of showing you the same thing. The direction of the action potential's apparent movement is toward the right. This would be the soma axon hillock. And on the other end, we would have the terminal boutons. Okay, so the message is going to move one way. That's in the normal situation. I'll talk about a pathological situation in a second. So if we look at the membrane potential, we'll say that we've gotten a little bit of propagation. Here the voltage-gated sodium channels are open, sodium's coming in, causing a depolarization. Now, if we look, that sodium is going to leak both ways. When we reach these channels that are just closed but ready to respond, if we exceed threshold, they're going to open up and sodium's going to come in, like I said. To the left, though, toward the cell body, these are still in the refractory period. Okay, so these are inactivated. So at this point, because the sodium is leaking in and depolarizing the membrane, you would expect those channels to open. However, because they're in the inactivated or refractory state, they don't. When they become closed, when they reset and they're ready to go, by that time, the membrane has repolarized and we're below threshold. Okay, so the timing of these events is quicker, is very important for you to pay attention to. Okay, so as I said, sodium comes in, leaks both ways. These open because they're closed and ready to go. These, nothing happens. And then the membrane potential will reset. Okay, so here the membrane hasn't reset, but it will in a second. If we look at the next millisecond, that's exactly what's happened. So you can see that here, those channels that were closed but ready to respond open up. Sodium leaks in. The channels that were open have become inactive. So even though we're above threshold, nothing can flow. These don't open. or I should say, they don't allow the flow of sodium into the cell. Therefore, nothing happens. The ones that were inactivated before, but not ready to go, the membrane has repolarized, and now they're closed and ready to respond. Okay, and this is going to go all the way down the axon to the terminals. In a vertebrate axon, most of our axons are myelinated. Okay, so you know myelin is produced by oligodendrocytes. It's got important functions. If we look at the axon, it's important to note that at the node of Ranvier, this is the only place where we have ion channels. So the voltage-gated sodium channels and voltage-gated potassium channels are only at the node of Ranvier. Nowhere else. They're not underneath the myelin. Myelin is going to function as an insulator. And again, in this class, we're not going to go through the physics of it, but basically it prevents the depolarization from dying off. Okay, so even though ions aren't entering underneath the myelin is going to make it so that when sodium comes in, the depolarization is going to go from one node to the next. It's going to move to the right without decaying very much. So sodium comes in, I get a massive depolarization. Underneath the myelin, that signal is going to be carried. So when we hit the next node, we're above threshold. If we're above threshold, what happens? Voltage-gated sodium channels open, and sodium is going to come into the cell. Okay? So that's what the difference between the top and the bottom diagram is. Here's, again, sodium has come in. That current is going to be carried. That depolarization is going to be carried underneath the myelin. When we hit the next node, it's above threshold. Sodium comes in, recharges the signal, and that's going to happen again, and so on and so on. Without myelin, if somehow this were gone, that charge, the depolarization we got up here, would decay very quickly. And it would decay so quickly, in fact, that by the time we got to the next node, we might not be above threshold. So let's say we hit threshold here, sodium comes in, and then our signal decays so much that when we hit this node, we're below threshold. If we're below threshold, what happens to the voltage-gated sodium channels? Nothing, which means your signal dies. It just stops. You can imagine already why that's bad. Now, it turns out that when you have to use all those channels all the way down, conduction is actually fairly slow. So if we compare the conduction velocity, how fast we go from the axon hillock to the terminals, it's quicker in myelinated axons. Okay. Unmyelinated axons, as it says here, are relatively slow compared to myelinated axons. Okay, I'll just show you a couple quick things. This has been determined empirically, so experiment after experiment showed this. Some geeky neuroscientists, you'd have races, one with a myelinated, one with an unmyelinated axon, and to see whose signal went faster, things like that. But we know for a fact that it is slower. So we'll present a stimulus, 1 millisecond, it's propagated a little bit, 2 a little more, 3 a little more. Okay, this isn't meant to be an absolute, but you can see this would be the slope. In a myelinated axon, though, it's much, much quicker. So 1 millisecond after, we've already gone to the next node. Okay, so you can see that that distance is much greater. 2 milliseconds, the next node. Okay, and so on. So you can see that if we look at the same amount of time, just because of the physics, it's going to be much, much faster in a myelinated axon. Again, just another illustration of this principle. One thing I do want you to know, humans have one type of unmyelinated axon that is of huge clinical relevance. You don't need to remember this, but they're so-called C fibers. These carry pain information. So these are the main unmyelinated axons in your body. Everything else you can assume is myelinated. But if we're talking about pain, it is carried by unmyelinated axons. And some of this you can see is slow. If you put your hand on a hot stove, very often you'll move your hand and realize you've moved before that pain, before the hurt information actually makes it to your brain. And part of that is because pain information is unmyelinated axons. Everything else will be carried by myelinated axons. So after this video, hopefully you can define propagation and you understand why it's important. Saltatory conduction. I did not use that word in the video, but saltatory conduction is jumping from node to node. Okay, saltatory has to do with jumping. So when we talk about how we move in a myelinated axon, the fancy name for that type of propagation is saltatory conduction. And then finally, what would happen in demyelinating disorders? This is a thought question for you to think about, and this is something that we'll have some problem sets in class where we'll go over this.